Hey guys, so hopefully this week you have really gotten a lot from all of the engage and explore activities, and up to this point you should have completed all the way up through explain one cell division KWL chart. So hopefully you have that chart in your notebook, and you found a lot of really good information in the cell division of complex organisms Stemscopedia, and watch these really good videos about mitosis, which is cell division, and cell differentiation. Okay, make sure you watch both of those. Now today what you're going to do is you're going to use the Stemscopedia once again. That's really, think of that as almost like our textbook. Okay, that's where all of our really good information is, of just explaining everything in detail. And what I've gotten is just to make sure that everyone is good to go for your quiz, which is coming up soon. You have some questions built into this to test your knowledge. Now, this is towards the end of this topic, and you also have all of this information available to you in order to answer these questions, because you're going to use the Stemscopedia to answer the questions. So I will be grading these on accuracy, not just effort, not just participation. They will be graded whether you got the questions right or got them wrong. Okay, so make sure you're paying attention and you're putting a lot of effort into this to get the answers right. So again, here's the Stemscopedia. You've seen some videos walk you through this, but you haven't had me walk you through it yet. And some of you may glean more from me than the video. We'll see. So I just want to walk you through this. So there's two really big concepts to this topic. One is that every single cell divides into two cells. That's how cell division happens, okay? That's how you started, as one zygote, one single cell, one sperm fertilized egg, and now you've grown into this complex organism. It's because that cell billions of times now, maybe trillions of times, has divided through this process. So let's talk about the whole cell cycle. First step is interphase. A cell will spend about 90% of its lifetime in interphase. So if a cell is just kind of chilling, doing its thing, doing its job, your skin cell is just kind of sitting there as a skin cell. If your liver cell is doing its job as a liver cell, that means that it is sitting in interphase right now, okay? It has three different branches. You've got G1, S, and G2. In G1 phase, this is where a cell differentiates into a specialized cell. So it starts as kind of what we call a stem cell, and then if it needs to be a liver cell, that's when it will develop into like a mature liver cell. If it needs to be a skin cell or an eyeball cell or whatever, a heart cell, stomach cell, this in G1 phase is when it kind of grows up and does its job. Okay, that's when it differentiates and matures into a functioning cell. S phase stands for synthesis. This is where DNA replication happens. Remember all the way back to that first gizmo in the first week of class with the introduction to gizmo activity, you guys actually modeled with that gizmo simulation the process that happens in S phase. All of the DNA within one cell gets split and copied. Now, why might it want to do that? Why is it so important that before a cell divides that the DNA is copied? Well, all of your cells have a certain amount of DNA. That DNA has the instructions to make the proteins that then tell that cell its job. Okay, think of like the proteins as like the worker in a factory. Okay, so if you have one cell that has all the instructions for how to make the proteins necessary for that cell to function correctly, let's say a skin cell, for example, okay, and then you take that cell and you divide it in half without duplicating the DNA, that means one of the new cells only gets half of the instructions and the other new cell only gets half of the instructions. Neither one has the full picture. Neither one has the full set of instructions for how to function properly. So neither cell would survive. So in S phase, your DNA duplicates. You get a duplicated copy of every DNA strand. That way, when mitosis happens, one of those copies goes into one new cell, so it has a full set, and the other copy goes into the other cell so that it has a full set, okay? That's what happens in S phase. It's super important so that each of the new cells has the same genetic information. All right, then a cell goes into G2 phase. This is where the cell is really kind of preparing to go through mitosis, preparing to go through cell division. So some of the other structures inside of the cell called organelles are going to start to duplicate. The cytoplasm, the fluid inside of the cell, is starting to gain in volume so that everything is ready to go for those two cells to split apart. Okay, so G1, the cell grows up and does its job. S is where the DNA replicates. G2 is where the cell is really preparing for mitosis. Now, there is an exception called G0. Some cells do not need to divide. So some cells like mature cardiac muscle cells and mature neurons they enter what's called G0 phase. They go to G1 and then G0, so they don't go through S or G2 ever again. Okay, and here it walks you through in detail G1, S, and G2. Once that is over, mitosis happens. Okay, so mitosis, I always remember this, and maybe say this out loud with me while you're watching this video. PMAT, 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 PMAT. No, not what your dogs use. It's an acronym for PMAT, prophase, metaphase, anaphase, and telophase, the 4 steps of mitosis. Okay, so in prophase, this is where the DNA condenses into chromosomes. They kind of wind up into chromosomes, and that is so that they're really easily packageable and movable throughout the cell. They're not big, long spaghetti strands. They're little packages of chromosomes. Metaphase is where the chromosomes then line up in the middle of the cell, okay? And there's a reason for that. So metaphase, middle, metaphase, middle. Think about that. Anaphase is where the spindle fibers that are attached to the chromosomes that are lined up in the middle of the cell pull the chromosomes apart, pull them in half, so that half of the DNA goes to one side of the cell and half the DNA goes to the other side of the cell. Okay, there's a point for that. Anaphase, or I just went through anaphase. Telophase is where the spindle fibers break down. Basically, it's the opposite of prophase. The DNA starts to relax, and the two cells, now the cell is kind of elongated, and it starts to find, telophase, it starts to split into 2 different cells. So that's why the DNA is on either end, so that when the cell splits, the DNA is where it's supposed to be. So telophase is it's the opposite of prophase. It's getting ready to split. Something called the cleavage furrow forms. It's like the little indentation where the cell is starting to split. That's in animal cells. And then cytokinesis happens right after mitosis. So mitosis is PMAT. The cell is doing everything right up to the point of dividing. Cytokinesis, which literally means cell movement. Cyto meaning cell, kinesis meaning movement, is just the action of the cell splitting. Okay, that's all cytokinesis is. Very simple. So here you can see a picture of all of those, interphase, and then PMAT, and then the 2 daughter cells after cytokinesis. All right, now how do we go from that to all of these super specialized cells? You have so many different kinds of cells all over your body you don't even know, like skin cells, liver cells, nerve cells, heart cells, eyeball cells, all those. Well, that is when cell differentiation happens. Okay, cell differentiation is when your DNA in those cells starts producing proteins, and it will produce the proteins needed within that specific cell. Hopefully you saw that acted out in Explore 3. Okay, so all of your cells have the same DNA, but not all of the same proteins are made in every cell. So for example, skin cells have the same DNA as your nerve cells, but proteins that give the skin cell its job as a skin cell are made there. And then the proteins needed for a nerve cell to do its job are made in the nerve cell, okay? DNA has all the instructions to make these proteins, but only certain ones of those are being read. Think of it like a cookbook. Think of your DNA is like a cookbook. You have the same cookbook in all of your cells, but only certain cells need certain recipes out of that cookbook. And a recipe is the instructions for how to make a protein, okay? So maybe if you're working in, like a lunch diner, you have a cookbook and you're only reading the lunch recipes out of that. But maybe, you're working somewhere and you're a pastry chef. You maybe have the same cookbook, but you're not reading how to make a hamburger. You're a pastry chef. You're reading how to make the desserts, okay? So same cookbook, same broad set of instructions, but certain cells need certain proteins to be made. Okay, I hope that makes sense. If there is an error when DNA is getting copied, that is called a mutation. We're going to talk about those a little bit later on. One of the biggest consequences that mutations can have in terms of cell division is sometimes it can cause G1 phase to become incredibly short. So whenever the P53 gene is mutated, the cell loses its ability to control how fast it replicates. And so what you get is cells are going through mitosis over and over and over and over and over way too fast. And you have this big bundle of cells that are just going through crazy amounts of unregulated cell division. We call that a tumor, okay? And those cells are called cancer cells. So that's actually how cancer is. It's a mutation that messes up how fast a cell does mitosis, all right? So without cell differentiation, this is really important. We could not have specialized tissues. All of our cells would be the same. So mitosis doesn't give us specialized tissues. It just copies the cell. It just duplicates the cell. Those specific proteins being made, given those cells their specific jobs, is what is called cell differentiation. And without that, we would not have any specialized tissue like heart tissue, stomach tissue, skin tissue, brain tissue, okay? I think I've covered really the main points of this. Hopefully I didn't rant for too long. So now go ahead and use the information in here in the Stemscopedia, answer those questions, turn that in for a grade. And that's simple. I won't call it easy, but it is a simple assignment today. Okay, good luck. If you have any questions, feel free to email me or call me and ask me. Okay, good luck, guys.